I'm John Weisel from the University of Pennsylvania in Philadelphia, Pennsylvania, and my research covers a variety of aspects of blood clotting with the primary focus on molecular and cellular mechanisms of fibrin polymerization, fibrinolysis, platelet aggregation, and clot contraction, and correlations with clinical implications. Russ Doolittle was certainly a leader in these fields when I began my career, so I knew of him and his work from the very beginning and met him at meetings very early on, including ISTH meetings. Russ Doolittle spent nearly all his career in the chemistry department at the University of California in San Diego, starting in 1965. He was an exceptional biochemist with an intuitive grasp of how the properties of amino acids translate into the structure and behavior of proteins. Amino acid composition and sequencing was the main component of his work for many years, to the point that he once said, only half in jest, that he intended to call his scientific memoirs Waiting for Arginine, since arginine is the last amino acid to elute from the sequencing chromatography column. His work began with fibrinogen, and he had an early hallmark publication on the evolutionary implications of the fibrinopeptide sequences from different species, which was one of the first experimental papers published on molecular evolution. At that time, before rapid DNA sequencing made protein sequencing obsolete, the determination of the amino acid sequence of fibrinogen's 3 polypeptide chains was a monumental task, carried out in competition with other prominent groups. That primary sequence and the identification of the disulfide bonds holding fibrinogen together provided many insights into fibrinogen structure and functions. Russ's knowledge of the properties of proteins, peptides, and amino acids also led to one of his most famous works outside the blood coagulation field, the Kite-Doolittle hydropathy scale. This scale is widely used for identifying hydrophobic regions in proteins, both surface exposed and transmembrane regions. After Russ determined the amino acid sequences of the alpha and beta chains of fibrin exposed upon cleavage of the fibrinopeptides, he realized that these sequences could provide the key to how fibrin polymerizes. Through studies with short peptide analogs of these newly exposed sequences, he discovered that they could be used to inhibit polymerization, leading to the concept of complementary binding sites that he called knobs and holes that are responsible for fibrin polymerization, basically for holding clots together. This is an example of his remarkable insight into protein structure, because he used this terminology long before determination of the crystal structure that demonstrated their nature. Russ officially retired early, but then continued to work and succeeded in crystallizing some large fibrinogen fragments through some clever biochemistry. He then stubbornly refused to hand over solving the structure to anyone else and taught himself practical crystallography, working with his students and a local crystallographer. The structure determination for such a large protein was quite difficult at that time. Again, the determination of high-resolution crystal structures of the entire fibrinogen molecule was an intense competition with several other groups. However, this work enabled a new phase in the research on fibrinogen, because it now became possible to understand many structure-function relationships. In his later years, the evolutionary origin of the blood clotting system became a grand unifying theme in his research, starting with fibrinogen, but then going on to include the entire coagulation cascade. He summarized this work in his 2012 book, The Evolution of Blood Clotting. Russ went on to theorize about more general evolutionary principles and became a public spokesman. Because the coagulation cascade is so complex, it is a favorite target for creationists and all those skeptical of evolution. As a result, Russ became involved in both printed and oral public debates on evolution, and thus became an advocate for science. For his early work, Russ was elected to the National Academy of Sciences in 1984, and he received many other honors throughout his career. Besides his influence on the field, Russ mentored quite a few students. He carried out his research with a series of graduate students and postdocs, and he was closely involved in their work. usually carefully going over the results with them at the end of each day. Russ could go on to great lengths to help other scientists, be they old friends, colleagues, or new acquaintances, by sharing his opinions and materials that he developed. I have heard from colleagues that he would go out of his way to set up visits for scientists who came to his lab, including locating housing and helping them in many other ways. At the same time, Russ was very competitive, and nearly all his colleagues could tell colorful stories of how far he would go to come out on top. Russ was a committed member of the ISTH and was awarded honorary membership in the society, which was given in recognition of his dedication and leadership within the society. He played many roles within the ISTH, but one of his greatest was through being president of the 1985 International Congress on Thrombosis and Hemostasis in San Diego, and being primarily responsible for organizing that meeting. In addition, Russ had a keen sense of humor and great storytelling abilities. Besides his dedication to science, he was an avid reader and movie buff, passions he shared with his wife of more than 60 years, Fran. They loved to travel together, often when Russ's work was calling him from around the world. In summary, Russ will be missed by his friends and colleagues, but his legend will live on.